Okay, let's jump right in. In this explainer, we're doing something really fascinating. We're zooming all the way out from the microscopic fragility of our DNA, right out to the life-changing clinical realities of ocular genetics. We're going to explore the intersection of molecular biology, clinical medicine, and patient counseling. You see, when we talk about the eyes, specifically the retina, the lens, and the optic nerve, we're looking at structures with incredibly strong genetic foundations. And understanding the mechanics behind these foundations, well, it doesn't just help doctors diagnose diseases. It actually empowers them to predict outcomes and ultimately fundamentally change patients' lives.
Now this quote right here, this perfectly captures the central mystery of our biology. Basically, a single gene mutation doesn't always lead to a perfectly predictable outcome. Scientists call this allelic heterogeneity. It basically means you could have two different patients with a mutation on the exact same gene, yet they present with completely different clinical symptoms. And on the flip side, you could have patients with the exact same symptoms caused by mutations on entirely different genes. It's this complex, beautiful, and, let's be honest, sometimes super frustrating puzzle that geneticists and ophthalmologists have to solve together.
So, to unpack this, we've broken our explainer down into three parts. First, the micro, single genes. Second, the meso, tracing the family tree. And third, the macro, guiding patients.
Let's start with part 1, the micro, single genes and the really profound impact of microscopic mutations. All right, so monogenic eye diseases. These trace back to an error in just one single gene. Think of your DNA like a massive, highly detailed instruction manual. If just one letter, literally one single nucleotide, is swapped or deleted, the resulting protein might be completely abnormal. It can start accumulating inside the cell like uncollected trash until it causes outright degeneration. It is absolutely incredible, and kind of scary, how one microscopic typo can cause such massive clinical problems.
Retinitis pigmentosa, or RP, is honestly the perfect real-world case study for this. It isn't just one disease, it's actually a whole group of inherited retinal dystrophies associated with over 200 different genes. What happens is the rods, those are the cells that handle your low-light vision, they degenerate first, which causes night blindness. As the disease progresses, patients experience a shrinking visual field. It's kind of like looking through a narrowing tunnel, and eventually it impairs their central vision entirely. But RP certainly isn't alone in this monogenic category. A single mutation can also disrupt the development of the eye's anterior segment, leading to congenital glaucoma, which is often linked to the CYP1B1 gene. Or a defect in just one gene responsible for melanin synthesis can cause albinism, which deeply affects ocular pigmentation and early visual development. We see this in juvenile macular dystrophies too, which unfortunately carry a pretty poor visual prognosis. In all these cases, it's like a single genetic domino falls, knocking over a highly specific physiological pathway.
Now, genetic diseases are rarely perfectly predictable. Take variable expressivity, for example. The exact same mutation can cause a really mild disease in one family member, but a severe, blinding disease in their sibling. It's the exact same error, but it expresses itself at different volumes, so to speak. Contrast that with reduced penetrance. This is where a person carries the exact genetic blueprint for the disease, but shows absolutely zero clinical signs. They are completely asymptomatic. This wild variability is exactly why taking a family history requires some serious detective work.
Moving on to part 2, the meso, tracing the family tree and mapping inheritance across generations. When doctors are evaluating suspected inherited diseases, there are 4 fundamental transmission paths they look for. Autosomal dominant requires only one mutated copy of a gene, meaning a child has a 50% chance of inheriting the disease, with males and females equally affected. Autosomal recessive needs 2 mutated copies. Usually the parents are completely healthy carriers here, but the disease often strikes earlier and harder. Then we have X-linked, where the mutation rides on the X chromosome, overwhelmingly impacting males. And finally, mitochondrial inheritance, which is passed exclusively down the maternal line and tends to strike tissues that are super power hungry, like the retina and the optic nerve. Let's zoom in on that autosomal recessive pattern for a second. If both parents are symptom-free carriers, there is a 25% chance a single child will inherit both mutated copies and be affected by the disease. There's a 50% chance they'll inherit just one copy, becoming a symptom-free carrier exactly like their parents. And there is a 25% chance they'll inherit 2 totally healthy copies and be completely unaffected. Visualizing these odds is absolutely crucial when doctors are explaining risks to prospective parents.
Bringing it back to our retinitis pigmentosa case study, the X-linked variation highlights a really important biological vulnerability. Because males possess an X and a Y chromosome, they just don't have a backup healthy X chromosome to rely on if the one they inherit carries a mutation. Females with two X chromosomes generally have that backup. Because of this, X-linked RP hits males much harder, resulting in significantly more severe symptoms and a much earlier onset of vision loss.
Now, you might be thinking that knowing these patterns makes tracing a disease super easy, but inheritance is messy. New mutations can just arise out of nowhere. A patient could literally be the very first in their bloodline to have the disease. Plus, family histories are often incomplete. Add in that phenotypic variability we talked about earlier, where mild cases and relatives go totally unnoticed, and you get a very blurry picture. So the vital takeaway here, a complete absence of family history absolutely does not exclude the possibility of a genetic disease.
And finally, Part 3, the macro, guiding patients through counseling and the clinical future.
Look, genetic counseling is definitely not just handing a patient a pamphlet and sending them on their way. It is a highly structured, step-by-step process. Step 1 is establishing a rock-solid diagnosis, often confirmed by genetic testing because without that, risk calculations are basically useless. Step 2 is building a detailed pedigree chart. Step 3 is the actual risk assessment based on those inheritance patterns. Step 4 involves identifying who else in the family might need a referral. And Step 5, which is perhaps the most delicate, is communication and ethics. Counselors help families adapt to these risks, maintain confidentiality, and make incredibly personal family planning decisions that align with their own values. Now, looking closely at Step 3, the risk assessment, there is this fascinating caveat. Standard recurrence risks, like that 50% coin flip for an autosomal dominant trait, can be deeply altered by penetrance. For instance, in retinoblastoma, which is a childhood eye cancer, a patient might inherit the dominant mutation, but because the penetrance isn't 100%, they might never actually develop the tumor. So a genetic counselor can't just throw raw percentages at a family. They have to contextualize the clinical danger by explaining exactly how penetrant that specific mutation actually is.
So why does all this meticulous mapping and counseling actually matter? Well, the clinical importance is deeply human. Understanding these genetic mechanics enables early diagnosis for at-risk individuals, sometimes way before symptoms ever even begin. It facilitates the screening of extended family members who might be totally unaware they're in danger. It provides families with the runway they need for long-term emotional and structural planning. And, perhaps most excitingly, pinpointing the exact genetic error unlocks the door to emerging, targeted gene therapies.
We are literally on the precipice of a medical revolution right now. Ocular genetics is evolving from a discipline of simply predicting blindness to actively reversing it at the DNA level. As we transition from just reading the human genome to actively rewriting it, we really have to ask ourselves, are we ready for a future where our genetic destiny is no longer written in stone? Keep questioning the microscopic world around you. Thanks for joining me on this explainer, and I'll catch you next time.